video is for educational purposes only. Uploading it in any social media platform is not allowed. Our topic for this week is all about microorganisms. Let us have our lesson objectives first. At the end of the lesson, you should be able to compare the structures of bacteria, protists, and fungi. Describe the impact of these microorganisms to living things including humans. And create a 3D structure of a bacteria. Did you know that microorganisms are very diverse? They can be found everywhere on Earth. They can live on land, in water, and in the air. They also be found in the bodies of all living things, like plants, animals, and humans. Microbiology is a field in biology that deals with the study of microorganisms. So, ang subject ng the study of microorganism is what we call microbiology. But the person who study the microorganisms is what we call microbiologist. Okay? Ang tawag sa ilaha, microbiologist. Now, Anton van Leeuwenhoek discovered the animalcules, so little animals that cannot be seen by our naked eye. That is why he discovered the microscope. Now, what do we call the disease-causing microorganisms? Call it pathogens. So, katong mga ang microorganisms nga makakause og sakit is called pathogens. Because in microorganisms, there are bad and good. Makahelp o makadaot sa atua. Okay? Now, it's time for us to discover what are the parts and function of bacteria.
Bacteria are prokaryotic cells that play an important role in human disease and health. They can cause disease, but are also part of the human microbiota and live on our skin, body, and on everyday objects in our environment. When compared to eukaryotic cells, the structure of bacteria is less complex due to a lack of nucleus and membrane-bound organelles such as mitochondria, endoplasmic reticulum, and Golgi complexes. Now, bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. Let's start with the cell wall, which is found on the outer surface of the cell membrane, and its most important role is to protect the bacteria. It consists of peptidoglycan, which usually helps differentiate whether bacteria are gram-positive or gram-negative. Gram-positive bacteria have a single thick layer of peptidoglycan above the plasma membrane, which allows them to retain the staining dye. And gram-negative bacteria have a thinner layer of peptidoglycan sandwiched between the surface membrane and the plasma membrane, so they can't retain the dye. Additionally, the cell wall helps maintain their shape. The round-shaped bacteria are called cocci. The rod-shaped ones are called bacilli. Spiral-shaped ones are spirilla. And sometimes the same bacteria can have multiple forms, in which case they're called pleomorphic. Some bacteria are covered by a capsule, which acts as a shield that protects the bacteria against phagocytosis and also helps the bacteria adhere to surfaces. The capsule is considered an important virulence factor since the strains that lack a capsule are less virulent. Underneath the bacterial cell wall, there's the plasma membrane, which is the most important layer because it encloses the cytoplasm, which is a gel-like substance composed mainly of water that also contains cell components, enzymes, and various organic molecules. If the plasma membrane is removed, the cell's contents spill into the environment, and the cell no longer exists. Now, the plasma membrane is responsible for most of the cell's relationship with the outside world, by acquiring nutrients and eliminating waste, and also maintains the interior of the bacteria in a constant, highly organized state. Usually, All plasma membranes are selectively permeable barriers, which allow certain ions and molecules to pass in and out of the cell while preventing the movement of others. However, in bacteria, the plasma membrane has other important roles, such as respiration, photosynthesis, and the synthesis of lipids and cell wall components. Now, in the bacterial cytoplasm, there are scattered cell components such as ribosomes, which are the sites of protein synthesis. These ribosomes can be found spread all over the cytoplasm, where they produce proteins that are destined to remain inside the cell, or they can be attached to the plasma membrane, and they are called plasma membrane-associated ribosomes. And in this case, they make proteins that will reside in the cell envelope or get transported outside the cell. Another structure found in the cytoplasm is the nucleoid, which is an irregularly shaped region that contains the cell's chromosome. The chromosomes of most bacteria are a single circle of double-stranded DNA, but some bacteria can have a linear chromosome, while others can have more than one chromosome, and these chromosomes contain the genes needed to provide instructions for protein synthesis in the cell. Additionally to the genetic material contained within the nucleoid, many bacteria contain extrachromosomal DNA molecules called plasmids. These plasmids are small, double-stranded DNA molecules that can exist independently of the chromosome, and they can be circular or linear. Plasmids have relatively few genes, and their genetic information is not essential to the bacterium, and cells that lack them usually function normally. However, plasmids often contain genes that confer a selective advantage to the bacterium, such as resistance to antibiotics. Finally, the bacteria can move or attach to other cells and surfaces with the help of structures that can be found on their external surface. So they have pili, which are hair-like structures that allow them to attach to other cells, and when these are shorter, they're called fimbriae, and help the bacteria attach to surfaces. They can also have flagella, which are long, whip-like protrusions that allow the bacteria to move. Alright, as a quick recap, the bacteria are prokaryotic cells that play an important role in human disease and health, and can be found almost anywhere in the environment. Bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. They have a cell wall which protects the bacteria, determines whether they are gram-positive or gram-negative bacteria, and also helps maintain their shape. Some bacteria can also have another extra layer on top of the cell wall called a capsule, which is a major virulence factor. Underneath the cell wall, there's the plasma membrane, which is the most important layer because it encloses the cytoplasm and defines the cell, having important roles such as respiration, photosynthesis, and the synthesis of lipids and cell wall constituents. The cytoplasm contains ribosomes, which are the site of protein synthesis, the nucleoid, which contains the genetic material, and plasmids, which contain genes that confer a selective advantage to the bacterium.
How does bacteria reproduce? So bacteria reproduce through sexually and asexually. Sexual method is through genetic recombination. So talagsa rin siya mahitabo. However, touch ni siya sa mga bacteria. Kaya nga naman, the parent bacteria produce new traits of bacteria. So, for example, nagsakit ka karon, nagamit kag antibiotic, next week, nagsakit na pud ka, same nga sakit, nagamit kag same nga antibiotic, dili na siya mamatay ang bacteria, dili na kagakaulian. Why? Because lahi na siya nga trait sa bacteria, mas likusgan siya kay nag-combine ang parent traits, okay, sa parent niya. So, nag-evolve siya. So, dili na siya madutlan sa katunga antibiotic. So, mag-change na pud ka og antibiotic. So, ingana ang sexual reproduction. In a sexual reproduction, there are three types: the binary fission, budding, and spore formation. When we say ang asexual reproduction is same trait lang ang ilahang maproduce, so dali na siya mamatay. Kaya kung unsa ang parent, maupod ang ihang daughter cell. Maugyod kay asexual man siya, wala siya combination of parent traits. So binary fission, for example, naputol ang bacteria, tubo gyapon to siya ang naputol. Tanawa ani. Kani siya naputol ni siya. So ang kaning naputol mahimo ni siyang daughter bacteria. Okay? And spore formation, nag-create siya og bud. So kanang bud, pag matanggal na siya sa parent, mahimo siyang offspring. So pareha gyud ang traits nilang duha. Wala siya na change. So kung nagsakit ka karon, same nga antibiotic, same nga sakit, mamatay dayon siya. So nga may rehahang chance of survival. Kung muhis ka tag bacteria class, dili ut pasabot nga puro bad ang bacteria. There are good and bad bacteria, okay? So what is the economic importance of bacteria? We have the food processing. So we need bacteria for our food processing. For example, kaning cheese, Yakult. Ang cheese is a combination of yeast and molds. That is why makaproduce taog cheese. Also, ang wine making. And ang Yakult. Diba? Very familiar. Kisa gayon mo Yakult. So, Yakult is na siya bacteria nga lactobacillus. So, lactobacillus is a good bacteria. Para na siya sa atong sabi. Para okay ka dyan. Next is fiber rotting. Kana siya is na siya bacteria nga Clostridium botulicum. So, kana nga bacteria class, mag-help na siya sa, for example, kaning lilies. Mag-help siya para mahimo na itong product. So, makapapag-on siya. So, na siya bacteria. Next is Escherichia coli or E. coli. Bacteria na siya class nga nasa atong large intestine or sa atong stomach. Mag-help siya og digest sa food. Kaya kung walay bacteria sa atong stomach, kung nagkaon ka rice, dili siya ma-digest, dili matunaw. So, ga-help siya. Next is tanning. Para ka ng mga skin sa animals nga himoong leather. Panit sa animals. Himoon siyang product, himoong bag, shoes. So, na yung bacteria dito. But, naapoy harmful effect of bacteria. Makakos siya og bacteria. Sa ganito mga microorganism nga makakos og disease, what we call them? They are called pathogens. Okay. So, we have the leprosy. Kanang leprosy na siya Mycobacterium leprae. Cholera. Diba kanang makuha na to sa mga hugaw na water. So, na bacteria nga Vibrio cholera. So, makaharm na siya sa atong body. Tuberculosis. Mycobacterium tuberculosis. Typhoid. Bacillus typhosus. So, kanang mga bacteria, some examples na siya class sa bacteria nga makaharm sa atua. So, not all bacteria are good and bad. So, what is protist? Protists are organisms nga makakause og disease. Naapoy, makahatag og benefit sa atua. So, we have here protozoans. Kaning protozoans is animal-like structure nga bacteria nga protist, pero makakos siya og disease. Tawag niya ay pathogenic na siya nga bacteria. Okay? Next is Plasmodium. Makuha na natin siya sa human blood cells. Bacteria na siya nga makadaot sa atuang blood cells. Through cause na siya sa malaria. Kasi kailaog lang mo ka malaria. Ang iyang genus name is Anopheles. So kung mapakad ka ana, ang iyang bacteria, Plasmodium, moadto na siya sa imuhang blood cells. Next is fungi. Ang fungi kay makatabang na siya sa atuang economy. However, naapoy mga harmful, okay? Makakos siya og ringworm. Kay bacteria man na siya. Naapoy mushroom, di ba ang mushroom? Kaya pwede natin siya makaon. Naapoy mga poisonous. So, be careful with that. So, for your activity, you are going to create a 3D model of a bacteria. So, 3D has 3D model. Okay, not just to create a 3D model of a bacteria, but also you will label the organelles. So, iyang mga parts, imuhang i-label. Mutahan ninyo og mga pangalan. So, you can create this by group. Maximum of 5 person. Okay? So, kung naamoy mga silingan dira nga pwede ikagrupo, so, pwede mo maggrupo. Okay? So, depende sa available materials ang inyong gamiton. Pwede clay, pwede styrofoam, pwede plastic bottle. So, depende sa inyong creativity. So, our grading rubric, appearance, 15 points. Kung 3D ba gyud siya, tsada, detailed, naabatanan nga organelles, ihang parts, naka-label, 10 points. So, in total of 35 points. So, you are going to pass it on November 8, 2021. So, don't forget to write your name in your model.
Thank you and God bless.